we go to the session exactly on time. The first presentation is Novel Biomarkers of Cardiovascular Risk and Unbiased Proteomic Approach. Dr. Peter Ganz, please. Phil?
Okay. Good morning. It's a real honor for me to be part of this symposium in honor of Dr. Meltzer. And thank you for inviting me to Israel. So I'm mainly known, if I'm known for anything, for studying biology of endothelial, of the human endothelium. But 4 years ago, I got involved in proteomics. And we haven't published anything yet. We are just writing our first papers. But I just wanted to give you an update of the field. So there's been a lot of hype about omics, and there are different types of omics listed here. They really haven't gotten us very far to date. But what I want to do is give you an update of one of these omics, which is the study of proteins, in our case, in blood, and this field is called proteomics. And we like proteins because unlike genes, which are static, so you're born with them and you really can't change them, or they don't change, the proteins integrate the effects of genes, the environment, and any medical therapies. And we know that for cardiovascular disease, the environment and the medical therapies can change outcomes which wouldn't be reflected in your genes. So I will focus specifically on two areas. One for sure, if I have time, I'll mention the other one. I'll talk about assessment of cardiovascular risk using proteins, and also how you can use proteomics to discover new mediators of cardiovascular disease. So let me start with the first one. So in proteomics, we measure proteins, usually just quantify their amount. And these are the available methodologies, the mass spec, the 2D gels, and ELISAs, antibody-based ELISAs. And they really are tedious methods with major limitations that I won't really summarize now. But we are using a completely new technology to quantify proteins. And this is a collaboration with a company in Boulder, Colorado called SomaLogic. We use aptamers to measure proteins. And what aptamers are short sequences of single-stranded DNA or RNA that bind to proteins in a similar way that antibodies would bind to proteins. And they have high affinity for proteins, similar to antibodies, but they are smaller than antibodies. And unlike antibodies, which have to be synthesized, typically by immunizing rabbits, these DNA fragments or short sequences can simply be synthesized. So how do these aptamers, these sequences of DNA, bind to proteins? So this is a crystallograph. Shown here is the protein, and in color is this sequence of about 40 or 50 nucleotides binding to the protein through two properties. One is based on charge. So you can see that the negatively charged phosphates, which are contained within each nucleotide, will bind to positively charged amino acids within the protein. But also we've modified a side chain to these nucleotides in the 5 position here with various potential side chains which allow for chemical bonding based not on charge but based on other characteristics, whether it's hydrogen bonding, hydrophobic bonding, or other types of bonds. So these are highly selective reagents. You just have to find the right aptamer for the right proteins. So how does the assay work? How do you actually measure proteins if you have these specific binding reagents, these aptamers, these DNA reagents? And so I'm going to illustrate this in this slide. Imagine somebody gives you a solution which contains 2 proteins, and one is 3 times as more abundant, shown in this yellow color, than the one shown in the green color. So this is an unknown solution for you. Somebody gives it to you, and what you do is you add aptamers, and we have aptamers now for over 3,000 proteins. So you add them, you don't know what's in the solution, but the right aptamer will bind to its cognate protein. So you can see 3 aptamers binding to this protein and another aptamer binding to its protein, and these aptamers are fluorescently tagged. as indicated by that star. And to quantify the amount of protein, we take advantage of the relationship in binding that one aptamer will bind to one protein. So what we do at this point is we dissociate the aptamers from the protein. And instead of quantifying the protein, we actually quantify the aptamer that had bound to the protein in a 1-to-1 ratio. And quantifying aptamers is very simple. It's DNA, and quantifying DNA by today's standards is something extremely simple. So we have a DNA chip which contains the complementary sequence to the aptamer. So these three aptamers will find their complementary sequence, and the amount of aptamer can be derived from the amount of fluorescence. And this aptamer will find on the DNA chip its complementary sequence, and again it can be quantified by the amount of fluorescence. So that the aptamer platform, this methodology, converts cumbersome protein quantification for much easier DNA quantification. And it's really a technology which I think will replace the other technologies that I mentioned earlier. And it's high throughput. We can do several thousand samples in about a week. It's all robotic now. So what do we do with this? So our initial aim was to see which proteins that we can detect in blood might relate to cardiovascular risk. And as you know, we already have some proteins that relate to cardiovascular risk, like C-reactive protein or NT-proBNP, but those were selected by a candidate approach. Here we would measure proteins in a fairly random way and use more of an agnostic approach. And so we wanted to identify individual proteins that relate to cardiovascular risk, and from the individual proteins then construct a risk prediction model, a multi-biomarker risk prediction model, and then we would determine whether this risk prediction model improves on risk discrimination beyond the available clinical risk engines. So we had two cohorts, a derivation cohort, which was 987 subjects. And this was a secondary prevention cohort, so they had stable coronary heart disease. And there was a San Francisco-based cohort where I work now. It's the so-called heart and soul cohort. And then we had a validation cohort, which was similar patients, stable angina patients. It was based in Norway. And one of its important characteristics is that it was that the quality of the blood samples was not quite as good. So they would kind of reflect the real-world blood sample quality that you would get outside the research setting. And we, at this point, even though we can measure about 3,000 proteins now, initially we measured 1,130 proteins in these blood samples. Both of these cohorts had at least 5 years of clinical follow-up. And our outcome was a composite endpoint of mortality, myocardial infarction, heart failure, and stroke. So what were the results? Well, when we screened 1,130 proteins, to our surprise, there are quite a few that statistically relate to cardiovascular risk to a Bonferroni corrected p-value of less than 0.05. But about 114 of these proteins associate with cardiovascular risk with what I would call a meaningful hazard ratio, the top quartile to the bottom quartile, that either exceeded 1.25 or was less than 0.75. And these are some of the examples of the proteins. So we ended up constructing a multi-biomarker risk prediction model from these proteins. So this was really work with past bioinformatics people. This is a complicated process. But I just want to illustrate some of the proteins that go into the model. As you can see, there are proteins, and these are shown as hazard ratios of association with cardiovascular outcome, hazard ratios of the top quartile to the bottom quartile. CRP is actually not one of our 11 proteins that we use to construct our risk prediction model. I just listed it here as kind of a gold standard that we are familiar with. These are unadjusted numbers. And you can see for CRP, as in many other publications in other cohorts, the risk ratio is about 2.39. But you can appreciate other proteins that are much more strongly associated with cardiovascular outcomes than CRP. So most of these cardiovascular risk predictors are newly described proteins, really pointing to the power of agnostic approaches, that we now find many proteins that we didn't realize had anything to do with cardiovascular disease. Now we can construct these risk prediction models. This is just data showing that if you look at the best quartile, the lowest risk quartile for outcomes in this risk prediction model, and you compare it to the worst quartile, there's about a 10-fold gradient of risk. So even though stable coronary heart disease is usually viewed as a fairly homogeneous condition, in fact, it's a highly heterogeneous condition, at least based on proteomics. In contrast, if you look at the Framingham secondary event score, which was published by D'Agostino some years ago, even if we change the coefficients to match our cohort, to best fit our cohort, really the risk prediction has a much smaller gradient risk. It's only about 2-fold. And if we use some of the kind of the standard approaches to risk discrimination, like the ROC curves, as in many previous publications, the secondary Framingham risk score really doesn't perform particularly well. The area under the curve about 0.66. And the proteins alone are better, area under the curve about 0.76. And when we add the two together, really proteins do almost as well as proteins plus the clinical risk factors. And in fact, if we do a backward selection modeling, so we have a model that consists of 11 proteins, and we try to add clinical variables to the proteins. It turns out that the model will accept age and sex, but it will really not accept any other clinical variables. So in the area of proteomics, clinical risk factors such as diabetes, lipids, smoking, or blood pressure no longer seem to matter for cardiovascular risk prediction. And so we do have a validation cohort. I'm not going to go through it. It very much confirms the derivation cohort. This is just examples of number of proteins and how they performed in the two cohorts. The derivation cohort is shown in black. The validation cohort is shown in red. And you can see that on the whole, the two cohorts actually give similar results. As I said, the validation cohort the samples were poorly handled. They were allowed to sit for 24 hours before the cells were separated from plasma. And that affected the performance of proteins derived from platelets or leukocytes. But nonetheless, on the whole, the validation cohort basically showed us the same thing as the derivation cohort. So I'm going to stop actually here. I'm not going to go to the second part of my talk. But question is, what's next? And I can tell you that as a biologist, what's next for me is understanding those 200 proteins or many more proteins that will come. And I'm totally fascinated with biology of many of them. But question is, how many proteins are there in blood? We measured 1,100 in this study. So really, proteins get into blood by one of two mechanisms. Some get there because you have necrosis in some organ in the body. And those proteins are really of not great interest to us because once we measure troponin, measuring additional proteins that basically illustrate the same pathway, it won't improve our risk prediction modeling. So we need proteins that reflect different pathways. And we think that about 2,200 proteins are secretable by cells in a regulated way because they have a signal sequence. They have a signal sequence that allows the cell to take the protein that's intracellular and turn it into an extracellular protein. That was published in Science in 2008. So currently, we can actually measure essentially all of those 2,200 proteins. So stay tuned. I think these models will improve, but I think they already perform in a way that makes me very, very optimistic that proteomics among all the omics is the correct way to go. So I'm going to stop here. Thank you very much for your attention.
Thank you very much, Dr. Ganz. We can take one or two questions, please. We have the time. Yeah. Can you please repeat it?
So MMP8, actually, all the MMPs are on our list of 1,130. I could show you the list. These were the ones that made it into the model. And to make it into the model really required two properties. One is that, the protein associated with cardiovascular risk. But the kind of modeling we do, it's based on, there are several different ways you could construct models statistically. There are the lasso techniques, the random, forest approaches, some Bayesian approaches. So this was based on a lasso modeling, and this is beyond my expertise. This is my team of bioinformatics people. But basically, the MMPs that I showed made it into the model, and MMP8 did not. And that's really based on just purely statistical considerations of constructing the strongest model.
Okay. Amir?
We will not allow a discussion.
Dr.
Shatan, please.
Congratulations. Fascinating. I'm not sure that I understood. You showed us a list of 11 or 14 proteins which are statistically significant. Let's say if we take 2 of them, 3 of them, and like in logistic regression, the others are still valid as independent?
I'm sorry, I couldn't...
If we take all the 11 and we take 3 of them, the strongest 3 of them, and if we put in the model the others are still valid as independent predictors, if I understand.
I'm sorry, so we constructed an 11 model, but you're saying could be reduced to model to fewer than 11? Was that your question?
We'll take the first 2.
We take the first 2. Yeah,
the others are still valid? If we...
The data are valid. So, there's a lot of debate how many proteins should go into the model.
Yeah.
And so if you kind of use these lasso approaches, it will give you several numbers. It will give you the optimal number of proteins, which is about 35. It will then give you what is called a parsimonious model, which is one standard error removed from that optimal number, which is 11. But frankly, if you construct a model that's only 9 proteins, it performs almost as well. So you probably don't need all 11. I would say when we publish this, it will probably be just a 9-protein model.
Well, thank you very much for this most stimulating presentation. And I am honored to present Philip Greenland from Chicago. I don't have to introduce him. He's worldwide known as one of the leading cardiovascular epidemiologists, and he will speak about the role of coronary CT and CTA in risk assessment. Phil, please.